Hello, my name is Mary Asmaty, and I am an undergraduate student in Dr. Chang's lab. And today I will be talking about the effects of bioinoculants in the microbial community of the soybean rhizosphere under drought conditions. Currently, the U.S. stands as one of the leading soybean producers in the world. And like any other agricultural product, the effect of drought has negatively impacted soybean yield and is predicted to increase in the next coming years due to climate change. Drought has been found to decrease the growth of beneficial proteobacteria in the soil, such as Bradyrhizobium japonicum, which is responsible for nitrogen fixation in the plant. Recently, bioinoculants, which are microorganisms that aid in plant growth, have been used as an alternative to chemical fertilizers to help improve crop yield. And the goal of this study is to examine the effect of these bioinoculants on the microbial community of soybeans under normal and drought conditions. We are specifically interested in three main components: the rhizosphere, which is the soil immediately surrounding the plant roots that harbor essential plant growth promoting bacteria, the bulk soil, which is right outside of this rhizosphere zone, and the root nodules, where the actual nitrogen fixation occurs. We collected samples from the Yoakum, Texas site, which is one of the drought-prone sites of a large-scale field trial being done in our lab. Rhizosphere soil and the near bulk soil samples were collected from 6 weeks point and harvest. The bulk soil samples were sent for physicochemical property evaluation. And for the microbial community analysis, DNA was extracted directly from soil samples, followed by PCR amplification of the 16S rRNA, which was sent out for sequencing. The reason that we use this 16S RNA gene is because it is commonly used in phylogenetic studies, since it has these highly conserved regions along with hypervariable regions that are used for prokaryotic classification and identification. Figure 3 here shows the principal component analysis that shows a difference in the physicochemical properties of irrigated bulk soil, indicated by the green color, versus the non-irrigated bulk soil, indicated by the red. So as we can see, the top half of the graph shows potassium, phosphate, and nitrate as prominent nutrients in the non-irrigated soil, whereas sodium and iron were found higher in the irrigated soil samples found below the x-axis. The length of the lines indicate the amount of variation the nutrient contributes to, and the distance between adjacent lines indicate the correlation between two different nutrients. So for the non-irrigated samples, we can see an association between potassium, phosphate, nitrate, and zinc, since they are very close to each other. And the sodium, iron, and calcium found on the lower half, they stand as unrelated variables contributing to the irrigated samples, since they all point out in different directions. For microbial community analysis, here in the top right corner, the alpha diversity is a measure of richness and evenness of OTUs, or operational taxonomic units. OTUs are groups of bacteria that share similar sequences, and figure 4 shows that about 7,000 sequences were randomly subsampled from each sample, which appears to be enough to capture most of the community diversity as indicated by the rarefaction curve approaching asymptote. The box plots on the right indicate a greater number of OTUs found in the irrigated samples, but shows no difference in the evenness of both irrigated and non-irrigated samples. The beta diversity presented in figure 5 measures the change in diversity of species under different environmental conditions. The findings suggest that significant diversity was indeed observed between irrigated and non-irrigated conditions. In conclusion, drought stress altered biotic as well as abiotic factors in the soil. The diversity of the microbial community was decreased by drought stress as observed by the number of OTUs and caused significant differences in beta diversity. This research will hopefully provide useful data for farmers in drought-prone regions, specifically in states like Texas. And we are further exploring the data and comparing 6 weeks harvest and nodule samples for future studies.